Welcome to our exploration of the remarkable world of cyanobacteria. Often referred to as blue-green algae, these photosynthetic bacteria are not just a footnote in Earth's history, they are pivotal players. Imagine a time billions of years ago when our planet's atmosphere was devoid of oxygen. It was these ancient pioneers who, through the process of oxygenic photosynthesis, began to transform the atmosphere, making it hospitable for the myriad forms of life we see today. Cyanobacteria's journey is one of survival and adaptation. From freshwater lakes and oceans to the most extreme environments like hot springs and arid deserts, these microorganisms have thrived. Their ability to fix nitrogen, endure desiccation, and perform photosynthesis makes them indispensable in nutrient cycling and primary production across various ecosystems. But what exactly are cyanobacteria? Structurally, they are simple yet sophisticated. These unicellular organisms, sometimes forming colonies or filaments, lack membrane-bound organelles but possess unique features like thylakoid membranes for photosynthesis and gas vesicles for buoyancy. Some species even house specialized cells like heterocysts for nitrogen fixation and akinets for survival in harsh conditions. Their physiological capabilities are equally diverse. Cyanobacteria harness light energy to drive photosynthesis, converting carbon dioxide and water into organic compounds and oxygen. They also fix atmospheric nitrogen into a biologically usable form, enriching ecosystems with essential nutrients. Remarkably resilient, these organisms can withstand extreme conditions, making them true survivors. Reproduction in cyanobacteria primarily occurs through binary fission, but they also reproduce via fragmentation and spore formation. In nitrogen-limited environments, some species differentiate into heterocysts to ensure a steady supply of nitrogen. Ecologically, cyanobacteria are both beneficial and problematic. They form the base of many aquatic food webs and significantly contribute to nitrogen cycling. However, they can also create harmful algal blooms, producing toxins that threaten aquatic life, animals, and even humans. As we delve deeper into their biology, structure, physiology, and reproduction, we begin to appreciate the profound impact cyanobacteria have had on Earth's history and their ongoing significance in modern ecosystems. Join us as we uncover the secrets of these ancient pioneers and their role in shaping the world as we know it. If you enjoyed this video, please like, share, and subscribe to our channel for more fascinating insights into the natural world. Welcome to a journey into the fascinating world of biological clocks. Before we dive into the intricate details of circadian rhythms, let's explore a fundamental concept: photoperiodic time measurement. This is the ability of plants and animals to measure differences in day versus night length, known as photoperiod, and use that information to anticipate critical seasonal transformations, such as annual temperature cycles. Imagine a world where the length of the day triggers adaptive responses in higher organisms like flowering in plants, gonadal stimulation in animals, and even hibernation. It's a sophisticated timekeeping phenomenon that ensures survival and reproductive success. But what if I told you that this capability isn't limited to complex organisms? Unexpectedly, we have observed this remarkable ability in cyanobacteria, unicellular prokaryotes with generation times as short as 5 to 6 hours. When exposed to short winter-like days, these tiny organisms developed enhanced resistance to cold. This adaptation was mediated by the desaturation of membrane lipids and differential programs of gene transcription, including stress response pathways. Just like in higher organisms, this photoperiodic timekeeping in cyanobacteria required an intact circadian clockwork and developed over multiple cycles of photoperiod. This discovery reveals that photoperiodic timekeeping evolved in much simpler organisms than we previously appreciated, enabling them to mount genetic responses to stresses that recur seasonally. Now that we've set the stage, let's delve deeper into the world of circadian rhythms and uncover how they shape the lives of organisms big and small. We all know about the feeling of jet lag, that grogginess that comes from traveling across time zones. It's a common experience for anyone who has flown across the globe, leaving us feeling disoriented and out of sync with the local time. This happens because our bodies operate on a roughly 24-hour cycle called a circadian rhythm. This internal clock is deeply ingrained in our biology, influencing when we feel alert and when we feel sleepy. This internal clock regulates many of our bodily functions, such as sleep-wake cycles, hormone release, and even our metabolism. It ensures that our body temperature, digestion, and other vital processes are optimized for different times of the day. Circadian rhythms are found in a wide range of organisms, from humans to plants to even tiny bacteria. These rhythms are not just a human phenomenon, they are a fundamental aspect of life on Earth. These rhythms are essential for survival, allowing organisms to anticipate and adapt to the predictable changes that occur throughout a day. By aligning their activities with the environment, organisms can maximize their efficiency and survival. For example, plants use their circadian clocks to anticipate sunrise and optimize photosynthesis. This allows them to make the most of the available sunlight, enhancing their growth and energy production. But what about organisms that face more dramatic seasonal changes? How do they cope with the varying conditions throughout the year? How do they prepare for the challenges of winter or the abundance of summer? These organisms have evolved sophisticated mechanisms to adjust their internal clocks and behaviors in response to seasonal cues. It turns out that some organisms can even use their internal clocks to anticipate these long-term environmental shifts. By doing so, they can prepare for the harsh conditions of winter or take advantage of the plentiful resources available in summer, ensuring their survival and reproductive success. Cyanobacteria, often called blue-green algae, are microscopic organisms that inhabit a wide range of environments, from freshwater lakes to the Arctic Ocean. Despite their small size, they play a crucial role in the Earth's ecosystem. Cyanobacteria are photosynthetic organisms, meaning they can harness sunlight to produce energy. In fact, they were among the first organisms to evolve this ability, and they played a crucial role in oxygenating the Earth's atmosphere billions of years ago. These ancient organisms also hold secrets to understanding how life adapts to changing environments. My research focuses on how cyanobacteria use their internal clocks to anticipate seasonal changes, a phenomenon known as photoperiodism. To study how cyanobacteria anticipate seasonal changes, my team and I designed an experiment that we fondly call the Ice Bucket Challenge for bacteria. We exposed different groups of cyanobacteria to varying artificial day lengths and temperatures, mimicking the transition from summer to winter. We started by growing the cyanobacteria in conditions that simulated long summer days. Then, we gradually shortened the artificial day length and lowered the temperature to mimic the onset of winter. We also included a control group that remained in constant summer conditions. This experiment allowed us to observe how the cyanobacteria responded to these simulated seasonal changes and whether their internal clocks played a role in their adaptation. The results of our Ice Bucket Challenge were striking. We found that the cyanobacteria exposed to gradually shortening days were much better at surviving the simulated winter conditions compared to those kept in constant summer conditions. When we analyzed the cyanobacteria's gene expression patterns, we discovered that those exposed to shortening days had activated a suite of genes involved in cold tolerance and stress response. These genes were activated even before the temperature dropped significantly, indicating that the cyanobacteria were anticipating the winter conditions. The control group, however, remained unprepared for the sudden temperature shift and exhibited much lower survival rates. This finding highlighted the importance of the cyanobacteria's internal clocks in anticipating and adapting to seasonal changes. Our findings suggest that cyanobacteria possess an internal calendar that allows them to track changes in day length and anticipate upcoming seasonal changes. This internal calendar is likely linked to their circadian clock, which regulates their daily rhythms. By sensing the shortening days, the cyanobacteria can activate the necessary physiological changes to prepare for winter, such as producing antifreeze compounds or altering their cell membranes to withstand colder temperatures. This ability to anticipate environmental change gives them a significant survival advantage. The Ice Bucket Challenge experiment provides compelling evidence that cyanobacteria, despite their seeming simplicity, possess a sophisticated timekeeping mechanism that allows them to thrive in fluctuating environments. Section 6. Climate Change and the Microbial World. Our understanding of how cyanobacteria anticipate seasonal changes has important implications for predicting the impacts of climate change. As global temperatures rise and weather patterns become more unpredictable, the ability of organisms to adapt to these changes will be crucial for their survival. Cyanobacteria, as primary producers in many ecosystems, play a fundamental role in the food web and global carbon cycle. Understanding how they respond to climate change is essential for predicting the health and stability of these ecosystems. Furthermore, the mechanisms that cyanobacteria use to anticipate environmental change may provide insights into how other organisms, including plants and animals, might adapt to a changing world. Section 7. Unlocking the secrets of bacterial clocks. The discovery that cyanobacteria can anticipate seasonal changes raises many intriguing questions for future research. These tiny organisms, often found in aquatic environments, have developed sophisticated internal clocks that help them survive and thrive in varying conditions. We are just beginning to unravel the complex molecular mechanisms that underlie this ability. Scientists are delving into the intricate dance of molecules and genes that enable these bacteria to keep track of time so precisely. One key area of investigation is identifying the specific genes and proteins that allow cyanobacteria to sense changes in day length and translate that information into physiological responses. This involves studying the genetic blueprints and understanding how these elements interact within the cellular environment. Understanding these molecular pathways could lead to new strategies for engineering crops that are more resilient to climate change. By mimicking the natural resilience of cyanobacteria, we could develop agricultural practices that better withstand environmental stresses. Another promising avenue of research is exploring the diversity of photoperiodic responses in different cyanobacteria species. Each species may have unique adaptations that offer insights into how life on Earth has evolved to cope with changing light conditions. By comparing the mechanisms used by cyanobacteria adapted to different environments, we can gain a deeper understanding of the evolution and adaptation of circadian clocks. This knowledge not only enriches our understanding of biology, but also opens up new possibilities for biotechnological applications. Section 8. Future Directions Towards a Deeper Understanding. The study of photoperiodism in cyanobacteria is a rapidly evolving field, with new discoveries being made at an unprecedented pace. This research has the potential to revolutionize our understanding of biological timekeeping, shedding light on how organisms adapt to their environments and its role in environmental adaptation. By studying these mechanisms, we can gain insights into how life on Earth has evolved to cope with changing conditions. As we delve deeper into the molecular intricacies of bacterial clocks, we uncover the complex interactions that govern these processes. These discoveries can lead to new insights into the fundamental principles of life, revealing the hidden rhythms that drive biological functions. The knowledge gained from studying these ancient organisms is invaluable. It provides a window into the past, helping us understand the origins of life on Earth. This information can inform our efforts to mitigate the impacts of climate change, develop sustainable agricultural practices, and create more resilient ecosystems. Additionally, it can advance our understanding of human health, offering new perspectives on how our own biological clocks function. The future of this research is bright, with endless possibilities for groundbreaking discoveries. The potential for discovery is vast, and the implications for science and society are profound. By continuing to explore the secrets of bacterial clocks, we can unlock new solutions for a more sustainable and resilient future. This research not only deepens our understanding of life, but also equips us with the tools to address some of the most pressing challenges of our time. Section 9. Conclusion. The significance of anticipating seasons. The ability of cyanobacteria to anticipate seasonal changes highlights the remarkable adaptability of life on Earth. Even the smallest organisms possess sophisticated mechanisms for sensing and responding to their environment. Understanding how these ancient timekeepers function is crucial for predicting the impacts of climate change, developing sustainable agricultural practices, and advancing our understanding of biological rhythms across all life forms. As we continue to explore the fascinating world of cyanobacteria, we can expect to uncover even more secrets about the intricate relationship between life and time. The study of photoperiodism in cyanobacteria is a testament to the power of scientific inquiry and the importance of understanding the natural world around us. Long-lived plants and animals clearly regulate their physiology according to seasonal changes in day length to appropriately adjust their physiology. Jabber et al. Found that cyanobacteria can do the same even though individuals only live for a few hours, a time shorter than a single daily photoperiod. Exposure to short photoperiods characteristic of winter stimulated these cyanobacteria to adjust their membrane lipids and gene expression to accommodate cold conditions. These responses required a functional circadian clock. Thus, population-based sensing of photoperiod appears to have evolved early, perhaps as a refinement of stress pathways. L. Brian Ray, Pause 2S. The discovery that even the simplest organisms can adapt to seasonal changes adds a new layer of complexity to our understanding of biological clocks. It shows that the ability to anticipate and respond to environmental changes is a deeply rooted trait, essential for survival across all forms of life. As we delve deeper into the study of cyanobacteria, we not only learn about their unique adaptations, but also gain insights into the evolutionary history of life on Earth. This knowledge not only enhances our scientific comprehension, but also underscores the interconnectedness of all living organisms, reminding us of the intricate and delicate balance that sustains life on our planet. Pause 2S. Thank you for joining us on this journey through the microscopic world of cyanobacteria and their incredible biological clocks. If you enjoyed this video, don't forget to like, subscribe, and hit the bell icon for more fascinating insights into the wonders of the natural world. Until next time, stay curious. As we conclude our exploration of cyanobacteria and their extraordinary circadian clocks, we find ourselves at the brink of new scientific frontiers. The discoveries we've discussed are just the beginning. Researchers are now delving into how these ancient timekeepers can be harnessed for modern applications. Imagine a world where the principles of cyanobacterial clocks are applied to improve agricultural productivity, optimize biofuel production, or even enhance our understanding of human health. These tiny organisms hold immense potential to revolutionize various fields. One exciting area of research focuses on genetic engineering. Scientists are exploring ways to modify the circadian rhythms of cyanobacteria to better align with industrial processes. By fine-tuning these biological clocks, we could create more efficient systems for producing valuable compounds, such as biofuels and pharmaceuticals. Moreover, the study of cyanobacterial clocks extends beyond their practical applications. It offers profound insights into the fundamental nature of life itself. Understanding how these simple organisms anticipate and adapt to environmental changes sheds light on the evolutionary history of circadian rhythms across all living beings. As we move forward, the collaboration between biologists, chemists, and engineers will be crucial in unlocking the full potential of cyanobacterial research. The journey is far from over, and the possibilities are endless. Thank you for joining us on this fascinating journey through the world of cyanobacteria. If you enjoyed this video, don't forget to like, subscribe, and hit the bell icon for more deep dives into the wonders of the natural world. Until next time, stay curious and keep exploring. Photoperiod and photosynthesis. Cyanobacteria are obligate photoautotrophs, meaning they rely on light to drive photosynthesis, which provides them with energy in the form of ATP and reducing power for carbon fixation. Photoperiodism plays a crucial role in regulating the timing and efficiency of photosynthetic activity. Light-dependent photosynthesis. During the light period, cyanobacteria capture solar energy through photosystems embedded in their thylakoid membranes. The absorbed light energy is used to drive the photolysis of water, producing molecular oxygen as a byproduct. The electrons generated from this process are used to reduce NADP+ to NADPH, and ATP is synthesized through photophosphorylation. These energy carriers are then used in the Calvin cycle to convert CO2 into organic compounds. Effect of photoperiod length on photosynthesis. The duration and intensity of light during the day affect the amount of energy available for photosynthesis. A longer photoperiod increases the time cyanobacteria have to capture light energy and synthesize organic compounds. Conversely, shorter photoperiods limit the time available for photosynthesis, potentially leading to reduced growth and metabolic activity. However, cyanobacteria can adjust their photosynthetic machinery in response to changing light conditions, such as modulating the expression of genes encoding photosystem components and adjusting pigment composition to maximize light capture efficiency. Photoinhibition. Excessive light, particularly during prolonged periods of intense sunlight, can lead to photoinhibition, a condition where the photosynthetic apparatus, particularly photosystem 2, PSII, becomes damaged by excess excitation energy. Cyanobacteria have developed mechanisms to protect themselves from photoinhibition, including the synthesis of carotenoids and other protective pigments that dissipate excess energy as heat, and the repair of damaged PSII components. By understanding these intricate processes, we gain deeper insights into how cyanobacteria thrive in diverse environments and how their photosynthetic efficiency can be harnessed for various applications. This knowledge not only advances our grasp of basic biological principles, but also paves the way for innovative solutions in energy, agriculture, and environmental management. Stay tuned as we continue to unravel the mysteries of these remarkable microorganisms. Nitrogen fixation is another vital physiological process in cyanobacteria that is influenced by photoperiodism. Many cyanobacteria can convert atmospheric nitrogen, N2, into ammonia, NH3, which can be incorporated into amino acids and other nitrogen-containing molecules. This process is catalyzed by the enzyme nitrogenase, which is highly sensitive to oxygen and cannot function in the presence of high levels of oxygen generated during photosynthesis. To overcome this challenge, cyanobacteria have evolved an elegant solution: the temporal separation of nitrogen fixation and photosynthesis. During the day, cyanobacteria focus on photosynthesis, generating oxygen and energy for their cellular functions. At night, when photosynthetic oxygen production ceases, nitrogen fixation can occur in an anaerobic environment. This temporal segregation is controlled by the circadian clock, ensuring that nitrogenase remains active under oxygen-limited conditions. In filamentous cyanobacteria such as Anabaena, nitrogen fixation is additionally spatially segregated into specialized cells known as heterocysts. Heterocysts are thick-walled cells that create a microoxic environment suitable for nitrogenase activity. The differentiation of these heterocysts is also influenced by photoperiod, as light availability regulates the balance between photosynthesis and nitrogen fixation within the cyanobacterial filaments. Understanding these sophisticated mechanisms not only enriches our knowledge of cyanobacterial biology, but also provides insights into how these microorganisms can be harnessed to improve agricultural productivity and environmental sustainability. As we delve deeper into the interplay between photoperiodism and nitrogen fixation, we uncover more of the remarkable strategies these tiny but mighty organisms employ to thrive in diverse ecological niches. Join us next time as we continue to unravel the mysteries of cyanobacteria and their profound impact on our world. Cyanobacteria exhibit a dynamic response to changes in light availability by altering the expression of genes involved in photosynthesis, nitrogen fixation, and other metabolic processes. Photoperiodism influences the circadian regulation of gene expression, allowing cyanobacteria to optimize their physiology based on environmental light conditions. The circadian clock and light-sensing mechanisms in cyanobacteria regulate the expression of a large number of genes in response to photoperiod changes. Light-regulated genes include those encoding proteins involved in the photosynthetic apparatus, such as photosystem 1 and 2 components, carbon fixation enzymes, and pigments that absorb light. For instance, in Synechococcus elongatus, the expression of genes related to photosynthetic activity peaks during the light period, while genes involved in nitrogen fixation and respiration are upregulated during the dark period. This temporal regulation of gene expression ensures that cyanobacteria can efficiently manage their energy resources and cellular processes according to the availability of light. Understanding how gene expression is finely tuned by photoperiodism not only provides insight into the sophisticated regulatory networks of cyanobacteria, but also opens up potential applications in biotechnology and agriculture. By harnessing these natural mechanisms, we could develop innovative strategies to enhance crop productivity and environmental sustainability. As we continue to explore the intricate relationship between light and life, the story of cyanobacteria offers a fascinating glimpse into the elegance of nature's design. The growth rate of cyanobacteria is closely linked to photoperiod. A longer photoperiod provides more time for photosynthesis, leading to higher rates of biomass accumulation and cell division. Conversely, shorter photoperiods can slow down growth by limiting the energy available for cellular processes. Cyanobacteria also exhibit light-induced cell division, where cell division is synchronized with the light period. In Synechococcus elongatus, for example, Cell division rates increase during the day when energy availability is high, and the process slows down at night. Understanding the relationship between photoperiod and cellular growth is crucial for optimizing conditions in both natural and artificial environments. By manipulating light exposure, we can potentially enhance the growth and productivity of cyanobacteria, which can have significant implications for biofuel production, wastewater treatment, and other biotechnological applications. As we delve deeper into the rhythms of these microscopic life forms, we uncover the delicate balance they maintain with their environment, a balance that holds the key to unlocking new avenues in science and industry. Photoperiodism in cyanobacteria plays a crucial role in their ecological success across a wide range of habitats. Cyanobacteria are found in diverse ecosystems, from freshwater lakes and oceans to soils, deserts, and polar regions. The ability to sense and respond to photoperiod allows cyanobacteria to adapt to seasonal variations in light availability and maintain their role as primary producers and nitrogen fixers in these environments. One environmental concern associated with photoperiodism in cyanobacteria is the formation of harmful algal blooms, or HABs. Under favorable conditions such as increased nutrient availability and longer photoperiods during the summer months, cyanobacteria can proliferate rapidly, forming dense blooms that produce toxins harmful to aquatic life, domestic animals, and humans. The regulation of growth and toxin production by photoperiod underscores the importance of understanding cyanobacterial photoperiodism and managing and mitigating the impacts of HABs. As we continue to explore the intricate relationship between photoperiod and cyanobacterial behavior, it becomes evident that these microscopic organisms hold significant sway over their environments. By leveraging this knowledge, we can better predict and control ecological phenomena, ensuring healthier ecosystems and safer water resources for all. Photoperiodism in cyanobacteria profoundly influences their physiological processes, including photosynthesis, nitrogen fixation, and circadian rhythms. By responding to changes in day length and light intensity, cyanobacteria can optimize their metabolism, growth, and survival in diverse environments. The regulation of gene expression and cellular activities by photoperiod ensures that cyanobacteria can balance energy acquisition and metabolic demands according to the availability of light. Understanding the effects of photoperiodism on cyanobacteria is not only important for understanding their ecology, but also for addressing environmental challenges such as harmful algal blooms and nutrient cycling in aquatic ecosystems. As we deepen our knowledge of these microscopic architects of life, we uncover the profound interconnectedness of all living systems. Cyanobacteria, though tiny, play a monumental role in maintaining the balance of our planet's ecosystems. By appreciating their intricate dance with light, we can better anticipate and mitigate the impacts of environmental changes, paving the way for a healthier, more sustainable future. Thank you for joining us on this journey into the world of cyanobacteria. Stay curious, stay informed, and continue exploring the wonders of the natural world. Cyanobacteria, ancient microorganisms known for their role in oxygenic photosynthesis, continue to be a subject of intense scientific research. These prokaryotic organisms are unique due to their ability to harness sunlight, fix atmospheric nitrogen, and produce a wide variety of metabolites. They play a crucial role in global carbon and nitrogen cycles and have significant ecological, biotechnological, and industrial importance. With the growing challenges posed by climate change, energy crises, environmental degradation, and food insecurity, cyanobacteria have gained increasing attention for their potential in offering sustainable solutions. The future of cyanobacterial research holds vast promise in areas such as bioenergy, environmental applications, synthetic biology, and biomedicine. Imagine a world where cyanobacteria are harnessed to produce clean energy, reduce atmospheric carbon dioxide, and even produce essential medicines. The possibilities are endless, and the scientific community is just beginning to scratch the surface of what these remarkable microorganisms can achieve. As we move forward, interdisciplinary research and innovative technologies will be key in unlocking the full potential of cyanobacteria. By continuing to explore and understand these microscopic powerhouses, we can pave the way for groundbreaking advancements that could transform our world for the better. Thank you for joining us on this journey into the world of cyanobacteria. Stay curious, stay informed, and continue exploring the wonders of the natural world. As we look to the future, another groundbreaking area of research is the potential of cyanobacteria in medicine. These ancient microorganisms are not only vital to our ecosystem, but could also revolutionize the field of biomedicine. 1.1. Pharmaceutical production. Cyanobacteria possess the ability to produce a wide range of bioactive compounds, making them promising candidates for pharmaceutical production. Researchers are exploring how these microorganisms can be harnessed to synthesize antibiotics, antivirals, and other therapeutic agents. By genetically modifying cyanobacteria, scientists aim to increase the yield and potency of these medicinal compounds, offering new avenues for treating diseases. Imagine a world where life-saving medications are produced sustainably using sunlight and carbon dioxide. This vision is not far-fetched, as ongoing research continues to unlock the genetic secrets of cyanobacteria, paving the way for eco-friendly pharmaceutical manufacturing. 1.2. Cancer treatment. Cyanobacteria are also being studied for their potential in cancer treatment. Certain species produce compounds that exhibit anti-cancer properties, such as apoptosis-inducing agents and inhibitors of cancer cell proliferation. By isolating and enhancing these compounds, researchers hope to develop novel cancer therapies that are both effective and less toxic than traditional treatments. Future research will focus on understanding the molecular mechanisms behind these anti-cancer properties and optimizing the production of therapeutic agents. The integration of cyanobacterial compounds with existing cancer treatment protocols could lead to more targeted and personalized therapies, improving patient outcomes and quality of life. 1.3. Drug delivery systems. Another promising application of cyanobacteria in medicine is their use as drug delivery systems. Due to their small size and ability to penetrate tissues, cyanobacteria can be engineered to deliver drugs directly to specific sites within the body. This targeted approach could enhance the efficacy of treatments while minimizing side effects. Researchers are investigating how to modify the surface properties of cyanobacteria to improve their ability to carry and release therapeutic agents. Advances in nanotechnology and synthetic biology will be crucial in developing these innovative drug delivery systems, offering new hope for patients with chronic and hard-to-treat conditions. In conclusion, the potential of cyanobacteria in medicine is vast and largely untapped. As scientists continue to explore these remarkable microorganisms, we can look forward to a future where cyanobacteria not only sustain our environment but also play a crucial role in improving human health. The journey of discovery is just beginning, and the possibilities are truly endless. Thank you for joining us on this exploration of the incredible potential of cyanobacteria. Stay curious, stay hopeful, and continue to believe in the power of scientific innovation to transform our world for the better. As we continue to uncover the remarkable capabilities of cyanobacteria, one of the most promising areas of future research lies in their potential for renewable energy production. These ancient microorganisms, with their natural ability to convert sunlight into chemical energy through photosynthesis, are attracting significant attention for their role in biofuel production. Cyanobacteria can be engineered to produce biofuels such as ethanol, butanol, and even hydrogen gas. By manipulating their metabolic pathways, scientists aim to enhance the efficiency and yield of these biofuels, making them a viable alternative to fossil fuels. Imagine a world where our energy needs are met sustainably, with minimal environmental impact, thanks to the power of cyanobacteria. The process begins with the cultivation of cyanobacteria in large bioreactors, where they are exposed to sunlight and carbon dioxide. Through photosynthesis, these microorganisms generate biomass, which can then be processed into various forms of bioenergy. This not only provides a renewable source of energy, but also helps in reducing greenhouse gas emissions, contributing to the fight against climate change. Researchers are also exploring the potential of cyanobacteria in producing biohydrogen, a clean and efficient fuel that can power everything from vehicles to industrial processes. By harnessing the unique properties of these microorganisms, we can pave the way for a greener and more sustainable future. The integration of cyanobacterial biofuels into our energy systems holds immense promise. As we continue to refine the technology and scale up production, the dream of a world powered by renewable energy is becoming an achievable reality. The journey ahead is filled with challenges, but the potential rewards are transformative. Thank you for joining us on this exploration of the incredible potential of cyanobacteria. Stay curious, stay hopeful, and continue to believe in the power of scientific innovation to transform our As we stand on the brink of a new era in energy production, cyanobacteria offer a beacon of hope. These microscopic powerhouses can be engineered to produce a variety of biofuels, including ethanol, biodiesel, and hydrogen. Their natural ability to synthesize lipids and hydrocarbons positions them as key players in the quest for renewable fuels. Through genetic modification, researchers are enhancing the metabolic pathways in cyanobacteria to boost lipid production and improve the efficiency of biofuel synthesis. Imagine the overexpression of key enzymes in the lipid biosynthesis pathway, leading to a surge in fatty acid production, essential components for biodiesel. But the innovation doesn't stop there. Cyanobacteria have a unique capability that sets them apart: the direct production of hydrocarbons. Unlike many other biofuel-producing microorganisms, these tiny organisms can convert carbon dioxide into alkenes, which can be refined into gasoline or jet fuel. This transformative ability opens the door to cleaner, more sustainable energy sources. The future of biofuel research is focused on optimizing these pathways, enhancing the light harvesting efficiency of cyanobacteria, and developing cost-effective methods for large-scale production. The journey is challenging, but the potential rewards are immense. As we harness the unique properties of cyanobacteria, we move closer to a world where renewable energy is not just a possibility, but a reality. This new dawn in biofuel production heralds a greener, more sustainable future for all. Stay tuned as we continue to explore the incredible potential of cyanobacteria and their role in transforming our energy landscape. The future is bright, and the journey is just beginning. As we delve deeper into the realm of biofuel production, the synergy between bioengineering and cyanobacteria emerges as a pivotal force. Imagine a world where the fusion of cutting-edge genetic techniques and the natural prowess of cyanobacteria leads to groundbreaking advancements in renewable energy. In recent years, scientists have made significant strides in harnessing the power of synthetic biology to fine-tune the genetic makeup of these microorganisms. By introducing novel genetic circuits and regulatory elements, researchers can precisely control the metabolic pathways of cyanobacteria, paving the way for enhanced biofuel yields. One fascinating area of exploration is the integration of artificial intelligence with cyanobacterial research. AI-driven models can predict the most effective genetic modifications, optimizing the production of biofuels while minimizing resource consumption. This marriage of technology and biology holds the promise of revolutionizing the field, making biofuel production more efficient and sustainable than ever before. Furthermore, collaborative efforts across disciplines are accelerating the pace of discovery. Engineers, biologists, and chemists are working hand in hand to design innovative bioreactors that maximize light exposure and nutrient distribution, ensuring optimal growth conditions for cyanobacteria. These advanced systems are not only scalable but also environmentally friendly, reducing the carbon footprint of biofuel production. The journey to a sustainable future is a collective endeavor, and the potential of cyanobacteria lies at the heart of this transformative movement. As we continue to push the boundaries of science and technology, we inch closer to a world where renewable energy is abundant and accessible to all. Stay with us as we explore the uncharted territories of cyanobacterial research, uncovering the secrets that will shape the energy landscape of tomorrow. The synergy of bioengineering and cyanobacteria is not just a vision, it's the blueprint for a brighter, greener future. Hydrogen gas is considered a clean and sustainable energy carrier, and cyanobacteria offer a unique biological method for its production. Certain species of cyanobacteria produce hydrogen naturally, as part of their metabolic processes during nitrogen fixation. However, hydrogenase enzymes responsible for this process are oxygen-sensitive, limiting the efficiency of hydrogen production in oxygenic conditions. Future research in this area will likely focus on engineering oxygen-tolerant hydrogenases or developing strategies to spatially or temporally separate oxygen evolution from hydrogen production. Advances in synthetic biology could allow for the re-engineering of cyanobacterial metabolic networks to increase the efficiency of hydrogen production, enabling the development of scalable biohydrogen systems. The pursuit of harnessing cyanobacteria for hydrogen production represents a promising frontier in renewable energy research. By overcoming the current limitations, we could unlock a new, sustainable source of hydrogen, further reducing our reliance on fossil fuels. Stay tuned as we continue to explore the potential of cyanobacteria in transforming our energy future. The field of synthetic biology offers vast opportunities for the manipulation of cyanobacterial systems. With advances in genetic engineering tools such as CRISPR-Cas9, researchers can now precisely edit cyanobacterial genomes, paving the way for the creation of customized organisms with enhanced metabolic capabilities. Imagine a future where we can tailor cyanobacteria to produce not just hydrogen, but a variety of biofuels and biochemicals. By re-engineering their metabolic pathways, scientists can enhance the natural abilities of these microorganisms to convert sunlight and carbon dioxide into valuable products. This approach could revolutionize our energy systems, making them more sustainable and less dependent on fossil fuels. It's not just about energy. Synthetic biology can also help in developing cyanobacteria that can clean up environmental pollutants or even manufacture pharmaceuticals. The potential is immense, but challenges remain. Issues like genetic stability, regulatory hurdles, and public acceptance need to be addressed. However, with continued research and collaboration, the promise of synthetic biology and metabolic engineering in cyanobacteria could lead us into a new era of innovation and sustainability. Stay with us as we delve deeper into the fascinating world of cyanobacterial research and explore the limitless possibilities that lie ahead. One of the future goals of synthetic biology and cyanobacteria is the development of custom metabolic pathways for the production of valuable biochemicals. By inserting or modifying genes involved in the synthesis of natural products, cyanobacteria can be engineered to produce high-value compounds such as pharmaceuticals, nutraceuticals, and industrial chemicals. For example, cyanobacteria can be designed to produce terpenoids, which are valuable bioactive compounds with applications in medicine, fragrances, and biofuels. Additionally, metabolic engineering could enable the sustainable production of bioplastics and biodegradable polymers. Polyhydroxyalkanoates, PHAs, for instance, are a class of biodegradable plastics that can be synthesized by certain cyanobacterial strains. Future research will likely focus on optimizing these pathways to increase yields and reduce production costs, making bioplastics a viable alternative to petroleum-based plastics. Imagine a world where the everyday products we rely on are derived from sustainable sources, reducing our dependence on fossil fuels and minimizing environmental impact. The potential of engineering metabolic pathways in cyanobacteria is not just a scientific endeavor, it's a stepping stone towards a greener, more sustainable future. The journey ahead is filled with challenges, but the promise of innovation keeps us moving forward. Join us as we continue to explore the groundbreaking research that holds the key to unlocking a new era of biotechnological advancements. Enhancing the photosynthetic efficiency of cyanobacteria is another key area of future research. Although cyanobacteria are highly efficient at capturing light energy, they still face limitations in terms of carbon fixation and energy conversion. Researchers are exploring ways to improve the efficiency of the Calvin cycle, the primary carbon fixation pathway in cyanobacteria. This could be achieved through the overexpression of key enzymes such as rubisco, or by introducing alternative carbon fixation pathways from other organisms. Moreover, synthetic biology approaches could be employed to engineer cyanobacteria to utilize non-photosynthetic carbon sources, or fix carbon under low-light conditions, expanding their potential in diverse environments. Imagine a future where cyanobacteria are fine-tuned to thrive even in the most challenging conditions, contributing to carbon sequestration and helping mitigate the effects of climate change. The promise of such advancements not only lies in environmental sustainability, but also in the potential economic benefits. Enhanced carbon fixation could lead to higher yields of valuable bioproducts, making cyanobacteria an even more attractive option for various industrial applications. As we continue to delve deeper into the genetic and metabolic intricacies of these remarkable organisms, the possibilities seem limitless. With each discovery, we move one step closer to a future where cyanobacteria play a central role in addressing some of the most pressing challenges of our time. Join us as we unlock the secrets of photosynthetic efficiency and carbon fixation, paving the way for a greener, more sustainable world. Cyanobacteria's natural ability to thrive in various environments and their role in biogeochemical cycles make them ideal candidates for addressing environmental challenges. These microorganisms have the potential to be harnessed for bioremediation, where they can help detoxify polluted environments by breaking down harmful substances. For instance, certain strains of cyanobacteria can absorb heavy metals and other pollutants, making them invaluable in cleaning up contaminated water bodies. Moreover, cyanobacteria can contribute significantly to the reduction of greenhouse gases. By enhancing their photosynthetic efficiency and carbon fixation capabilities, we can leverage these organisms to capture and store carbon dioxide more effectively, playing a crucial role in mitigating climate change. The integration of cyanobacteria into agricultural systems as biofertilizers is another promising avenue. Their ability to fix atmospheric nitrogen can reduce the reliance on chemical fertilizers, promoting more sustainable farming practices. In coastal and marine ecosystems, cyanobacteria can help restore degraded habitats by stabilizing sediments and providing a source of organic matter for other organisms. Their potential in generating renewable energy, such as biofuels and hydrogen, further underscores their versatility and importance in the quest for environmental sustainability. As we continue to explore the myriad of ways in which cyanobacteria can be utilized, it becomes clear that these tiny organisms hold immense promise for creating a cleaner, greener planet. The future of environmental applications of cyanobacteria is not just a scientific endeavor, but a crucial step towards preserving our natural world for generations to come. Join us in the next chapter as we delve deeper into the innovative uses of cyanobacteria in medicine and beyond, showcasing their incredible potential to revolutionize various industries and improve our quality of life. As we stand on the precipice of unprecedented climate change, the quest for viable solutions has never been more urgent. Enter cyanobacteria, tiny but mighty players in the global carbon cycle. By capturing atmospheric carbon dioxide during photosynthesis, these microorganisms hold the key to a greener future. Imagine a world where engineered cyanobacteria are deployed on a massive scale to sequester carbon efficiently. Research is already underway to enhance their natural abilities, developing strains that can capture and store carbon more effectively. These bio-based carbon capture systems could be integrated into our fight against climate change, providing a natural and sustainable method to reduce greenhouse gas concentrations. But the potential of cyanobacteria doesn't stop there. These remarkable organisms could also play a pivotal role in restoring carbon balance in critical ecosystems such as oceans and wetlands. Through geoengineering efforts, cyanobacteria could be utilized to remove excess carbon dioxide from the atmosphere, helping to mitigate the severe impacts of global warming. As we explore these innovative applications, it's clear that cyanobacteria offer a beacon of hope in our battle against climate change. By harnessing their capabilities, we can take significant strides toward a more sustainable and resilient planet. Join us in the next chapter as we uncover the groundbreaking potential of cyanobacteria in medicine and beyond, revealing how these microscopic marvels can transform our world for the better. As we continue our journey into the remarkable world of cyanobacteria, we uncover yet another groundbreaking application: wastewater treatment and bioremediation. Cyanobacteria's ability to grow in nutrient-rich environments, including wastewater, positions them as promising candidates for bioremediation. Imagine a future where these microorganisms are at the forefront of wastewater treatment, removing excess nutrients such as nitrogen and phosphorus. This could prevent harmful algal blooms and eutrophication in our aquatic ecosystems, paving the way for healthier water bodies. But the potential doesn't stop there. Cyanobacteria can be genetically modified to detoxify heavy metals, organic pollutants, and other hazardous substances. Picture engineered strains thriving in contaminated environments, breaking down pollutants into less harmful compounds. This transformative approach could revolutionize the cleanup of polluted soils, water bodies, and industrial sites. As we explore these innovative bioremediation strategies, it becomes clear that cyanobacteria hold the key to a cleaner and more sustainable future. By harnessing their natural abilities, we can address some of the most pressing environmental challenges of our time. Join us in the next chapter as we delve deeper into the potential of cyanobacteria in medicine and beyond, revealing how these microscopic marvels can transform our world for the better. As we continue our journey into the remarkable world of cyanobacteria, we uncover yet another groundbreaking application: cyanobacteria in medicine and pharmaceuticals. These microorganisms produce a wide range of bioactive compounds, many of which have antimicrobial, antiviral, and anti-cancer properties. Imagine a future where the natural product diversity of cyanobacteria becomes a rich source of novel pharmaceutical agents. From antibiotics that can combat resistant bacteria to antiviral compounds that can help control pandemics, the potential medical applications are vast. Researchers are already isolating compounds from cyanobacteria that show promise in treating a variety of diseases, including cancer. The unique metabolic pathways of these microorganisms allow them to produce complex molecules that are difficult to synthesize in a laboratory setting. But the potential doesn't stop there. Cyanobacteria can also be genetically modified to produce specific therapeutic compounds in larger quantities. This bioengineering approach could revolutionize the pharmaceutical industry, making it easier and more cost-effective to produce life-saving medications. As we explore these innovative medical strategies, it becomes clear that cyanobacteria hold the key to a healthier future. By harnessing their natural abilities, we can address some of the most pressing health challenges of our time. Join us in the next chapter as we delve deeper into the potential of cyanobacteria in bioenergy and renewable fuels, revealing how these microscopic marvels can transform our world for the better. As we transition from the medical marvels of cyanobacteria, we delve into an equally critical domain, the discovery of new antibiotics. With the growing threat of antibiotic resistance, the urgency to find novel antimicrobial agents has never been more pressing. Cyanobacteria, with their diverse range of secondary metabolites, present a promising solution. These microorganisms have been shown to produce compounds with potent antibacterial activity, offering a glimmer of hope in the fight against resistant bacteria. Future research will undoubtedly focus on screening various cyanobacterial species for these valuable antibiotics. Scientists are keen on understanding the mechanisms of action behind these compounds, which could pave the way for groundbreaking treatments. Moreover, synthetic biology holds immense potential in this realm. By optimizing the production of cyanobacterial antibiotics, or even engineering new biosynthetic pathways, we can create modified antibiotic molecules with enhanced efficacy and reduced toxicity. In this quest, cyanobacteria stand at the forefront, embodying the promise of a healthier, more resilient future. Join us as we continue to uncover the extraordinary capabilities of these microscopic marvels, transforming our world one discovery at a time. As we transition from the medical marvels of cyanobacteria, we delve into an equally critical domain, the discovery of new antibiotics. With the growing threat of antibiotic resistance, the urgency to find novel antimicrobial agents has never been more pressing. Cyanobacteria, with their diverse range of secondary metabolites, present a promising solution. These microorganisms have been shown to produce compounds with potent antibacterial activity, offering a glimmer of hope in the fight against resistant bacteria. Future research will undoubtedly focus on screening various cyanobacterial species for these valuable antibiotics. Scientists are keen on understanding the mechanisms of action behind these compounds, which could pave the way for groundbreaking treatments. Moreover, synthetic biology holds immense potential in this realm. By optimizing the production of cyanobacterial antibiotics, or even engineering new biosynthetic pathways, we can create modified antibiotic molecules with enhanced efficacy and reduced toxicity. In this quest, cyanobacteria stand at the forefront, embodying the promise of a healthier, more resilient future. Join us as we continue to uncover the extraordinary capabilities of these microscopic marvels, transforming our world one discovery at a time. But the story doesn't end there. Cyanobacteria are also a promising source of anti-cancer agents. Several compounds derived from cyanobacteria, such as cryptophycin and curacin, have demonstrated significant anti-cancer activity in preclinical studies. Future research could focus on isolating and characterizing new cyanobacterial compounds with potential anti-cancer properties, as well as investigating their mechanisms of action at the molecular level. Moreover, metabolic engineering could be used to enhance the production of these bioactive compounds, making cyanobacteria a viable source for large-scale pharmaceutical production. Imagine harnessing the power of these tiny organisms to develop treatments that could save millions of lives. The potential of cyanobacteria in medicine is vast and awe-inspiring, pushing the boundaries of what we once thought possible. Stay tuned as we explore the depths of this untapped resource and its promise for a brighter, healthier future. As we transition from the medical marvels of cyanobacteria, we delve into an equally critical domain, the discovery of new antibiotics. With the growing threat of antibiotic resistance, the urgency to find novel antimicrobial agents has never been more pressing. Cyanobacteria, with their diverse range of secondary metabolites, present a promising solution. These microorganisms have been shown to produce compounds with potent antibacterial activity, offering a glimmer of hope in the fight against resistant bacteria. Future research will undoubtedly focus on screening various cyanobacterial species for these valuable antibiotics. Scientists are keen on understanding the mechanisms of action behind these compounds, which could pave the way for groundbreaking treatments. Moreover, synthetic biology holds immense potential in this realm. By optimizing the production of cyanobacterial antibiotics, or even engineering new biosynthetic pathways, we can create modified antibiotic molecules with enhanced efficacy and reduced toxicity. In this quest, cyanobacteria stand at the forefront, embodying the promise of a healthier, more resilient future. Join us as we continue to uncover the extraordinary capabilities of these microscopic marvels, transforming our world one discovery at a time. But the story doesn't end there. Cyanobacteria are also a promising source of anti-cancer agents. Several compounds derived from cyanobacteria, such as cryptophycin and curacin, have demonstrated significant anti-cancer activity in preclinical studies. Future research could focus on isolating and characterizing new cyanobacterial compounds with potential anti-cancer properties, as well as investigating their mechanisms of action at the molecular level. Moreover, metabolic engineering could be used to enhance the production of these bioactive compounds, making cyanobacteria a viable source for large-scale pharmaceutical production. Imagine harnessing the power of these tiny organisms to develop treatments that could save millions of lives. The potential of cyanobacteria in medicine is vast and awe-inspiring, pushing the boundaries of what we once thought possible. Stay tuned as we explore the depths of this untapped resource and its promise for a brighter, healthier future. The development of synthetic ecosystems involving cyanobacteria and other microorganisms is another exciting area of future research. Cyanobacteria can be integrated into microbial consortia with bacteria, fungi, or algae to perform complex tasks such as bioremediation, biofuel production, or nutrient recycling. By engineering these synthetic ecosystems, researchers could create microbial communities that are more efficient and resilient than individual organisms. Future research will likely focus on understanding the interactions between cyanobacteria and other microorganisms, as well as optimizing the metabolic interactions within these consortia for specific applications. Imagine a world where these synthetic ecosystems can clean our environment, produce sustainable energy, and recycle nutrients efficiently. The potential of these engineered microbial consortia is immense, promising a future where we can tackle some of the most pressing environmental and energy challenges. As we continue to unravel the mysteries of cyanobacteria and their interactions with other microorganisms, we edge closer to a more sustainable and resilient world. Stay with us as we explore the promising future of synthetic ecosystems and microbial consortia and the incredible potential they hold for transforming our world. As we conclude our journey through the fascinating world of cyanobacteria, we are left with a profound sense of their potential. The future prospects of cyanobacteria research are vast and hold great potential to address some of the most pressing global challenges. From sustainable biofuel production and environmental restoration to the discovery of new pharmaceuticals and the development of synthetic ecosystems, cyanobacteria offer a versatile platform for innovative solutions. Advances in genetic engineering, synthetic biology, and metabolic engineering will continue to drive progress in these areas, unlocking the full potential of these ancient microorganisms for the benefit of society and the planet. As we harness the power of cyanobacteria, we pave the way for a more sustainable, healthier, and resilient future. The journey of discovery is far from over, and the promise of cyanobacteria continues to shine brightly on the horizon. Thank you for joining us on this exploration. Stay tuned as we venture further into the unknown, uncovering the incredible capabilities of these microscopic marvels and transforming our world one discovery at a time. In summary, cyanobacteria research holds the key to groundbreaking advancements in multiple fields. Their potential to revolutionize biofuel production, medicine, and environmental sustainability is immense. Together, we can unlock the secrets of these ancient organisms and create a brighter future for all.